Welcome all to his Pharmacology Difficult podcast. I'm your host, Dr. Radhika Vijay, MBBS MD Pharmacology, and this is the audio hub to get the best simplified basic tips, strategies, methods, and lots of ideas to learn better, understand better, and make your concepts crystal clear. If you really find and if there's a question hovering in your minds, is pharmacology difficult? Lend me your ears for a while and let in the magic of knowledge. Let's continue with our drug discovery process. So we had covered up to the structural and the physico-chemical properties of the drug molecules. Further, we're going to talk about the lead. Now, what is the lead? What do you understand by the term lead in this drug discovery process? Now, lead is a compound from a series of related compounds. It has some of the desired biological activity. It can be modified, it can be characterized to produce another molecule with a better profile. That's the advantage of having a lead molecule or a lead compound. That's what we call. It has all the desired biological activities and the abilities, and it can be modified enough and processed to form a molecule of a better profile of desired profile. Lead compounds are identified by different kind of methods. First of all, very common thing, serendipity. Yes, that's very common in pharmacology. From existing drugs, it can be identified. From some kind of exogenous sources, from some kind of endogenous sources. Then by rational drug designing, it can be formed or identified. And by combinatorial chemistry. And also with the process of HTS, that is high throughput screening. It can be identified with the advanced technologies like genomics, proteomics. Pharmacogenomics is a very important process and technique which finally leads to the lead compound discovery. So these are the methods and the different sources of the lead compounds, how they are discovered, where they are found, and all these kind of things. Now, serendipity means unexpected and very bizarre kind of discovery of a lead compound. It is not done with an effortful or intentional manner. One of the wonderful examples of this serendipity is the Alexander Fleming. He was working for the examination of systemic fluids and he accidentally observed Penicillium notatum. This is one of the most famous examples of serendipity. So many drugs have been identified in psychotic disorders also, and all these kind of. So serendipity led to the discovery of not only penicillin but also of many other drugs. Diethylamide, meprobamate, chlorpromazine, etc. From existing drugs, lead compounds can be identified. Many companies, they use established drugs from their competitors and they are used as a lead compounds. They are further modified and they are produced as better drugs with a better drug profile to be produced into a enhanced and better lead compound. A good example is original penicillin was not so selective, but the modern penicillin is quite selective, and this is a better drug profile. Coming on to the various exogenous sources and endogenous sources. Exogenous sources can be plant kingdom. Many drugs, are derived from exogenous sources like plant kingdom, like morphine, digitalis, curine, cocaine, nicotine, etc. And then they further lead to the development of other compounds which are developed synthetically, like local anesthetics are produced from cocaine. And similarly, we have endogenous sources of the drugs also. Endogenous source means natural ligand for the receptor. They are the sources. Like one of the good example is histamine. Histamine was used as a lead compound. And what did it was used to develop into? Cimetidine. That is H2 histamine antagonist. So histamine was the lead compound and then it led to the discovery or the formation of cimetidine. Some are examples also. Natural endogenous sources can be enzymes also. Enkephalin, they are the sources to produce enkephalinase inhibitors. So this is how the natural enzymes or natural ligands for receptors, they are modified and further developed or further identified to produce certain kind of antagonists, etc. Now from microorganisms, the various sources are there. Examples are cephalosporin, streptomycin, aminoglycosides. From marine sources also lead compounds are identified and developed. Discodermolide are one of the compounds and drugs from the marine sources. Another drug example bryostatin, cephalostatin. These are all developed from the marine sources. These are the anti-tumor agents that are developed from the marine source. Then we have lead compound from the animal sources. Antibiotic polypeptides, they are derived from the animal skin, like from the African frog. These drugs are from the skin of the African frog. These drugs are derived. Then we have lead compound identification from venoms and toxins. Teprotide. It is isolated from a Brazilian viper. It was a lead compound for the development of anti-hypertensive agents like captopril, etc. Yes, this is a fact. Then combinatorial chemistry can be one of the sources of the lead compounds, and the best process under this is the HTS, that is high throughput screening. So HTS, let's talk about it. HTS has been developed over the last 10 to 20 years, and it includes different kind of processes and procedures. I'm gonna list a few of them. Microplate activity assay, and the result of this assay is the enzyme inhibition, and the change in the color is there, which enables the identification of bioavailability. That is the procedure of microplate activity assay. Another procedure, gel diffusion assay. Biological target is mixed in soft agar, and while the process of the diffusion, a proper developing agent is sprayed upon the agar surface, and then in different zones the bioactivity it happens. Next procedure, one of the procedures is affinity selection assay. This is a kind of combinatorial chemistry. It's a cost efficient method. A compound library is applied to a protein target receptor, and one of these microplates, they are widely used. So these are for some of the procedures that are done under the high throughput screening methods, some of the assays. Now let's talk about the advanced technological processes like genomics and proteomics. They serve how to identify the lead compounds. Now genomics is to be taken one step further, and then it helps in the identify compounds which can be used for the treatment of specific diseases, and new drugs can be developed. What is proteomics? Genomics, if we define, what is genomics? Genomics, if we define, in this the drug discovery requires linking of the proteins to specific genes. That is the basic procedure of genomics. On the other hand, proteomics, it is a kind of molecular biological discipline in which the structure and the function of all the active proteins, they are encoded in a particular genome. Then another technique, DNA microarray technique. It's a technology, latest technology, in which a specific mRNA in an individual cell, it correlates with a specific protein. And then last but not the least, bioinformatics and cheminformatics, they also help in the lead compound discovery. In this, a particular knowledge of the discovery and the pattern recognition algorithms is applied to the genome and the proteome also, and they are engrossed with the experimental data, and then they help in drug designing. Pharmacogenomics is also very helpful. It's a new concept which helps in the target identification and the drug development. It is actually the future of the lead compound discovery, identification and development. If we talk about the conventional drug designing, they can be designed to, the drugs can be made to treat particular diseases. Single nucleotide polymorphism, that is abbreviated as capital SNP. It is very crucial to have individualized drug designs. So these are the different procedures and the techniques and the latest technological advancements in order to identify, in order to characterize and develop, and really which are the sources of the lead compounds generation, development and modification, all in all to have a better drug profile. I hope this information today was quite useful. We'll be covering more aspects in the upcoming episodes. For all the updates and latest episodes of my podcast, do visit www.ispharmacologydifficult.com, where you can also subscribe to my e-newsletter, that is Pharmacology Further e-newsletter. If you are listening to this podcast for the first time, don't forget to subscribe on whatever platform you are consuming this episode. Stay tuned. Stay safe, stay happy, stay enlightened. Thank you.